Hello, my name is Roger Watson and welcome to this session on the nervous and sensory systems. Before we start, I'd really encourage you to look at this Bozeman Biology YouTube link on the nervous system. The nervous and sensory systems are comprised of the central nervous system and the peripheral nervous system, and also the special senses. First of all, we'll look at the central nervous system, and the components of the central nervous system are the brain and the spinal cord. And here we see a side view and a back view of the brain and the spinal cord, showing you how they are related and in fact connected to one another. And in the side view, you'll note that the spinal cord is not straight. This diagram shows more detail, shows more parts of the brain, which we will come back to, and it shows the components of the spinal cord, which are the vertebral column, which are bones that we'll come to when we look at the skeletal system, and running down that spinal cord. The peripheral nervous system is everything else which is not part of the central nervous system. These are the nerves which extend from the central nervous system out to the rest of the body. Now, this is a rather busy looking diagram of the brain, but the different colours show very nicely the different lobes of the brain. There's a frontal lobe, and behind it a parietal lobe, at the back of the brain an occipital lobe, and then below the frontal and parietal lobes and in front of the occipital lobe a temporal lobe running along the side of the brain, and below that the cerebellum. And then you can see the medulla, which connects the brain to the spinal cord. You can also see here the central sulcus marked here. You can't see it in this diagram, we'll show it in a subsequent diagram, but if you look at the top of the brain, there's a deep fissure running from front to back, and that divides the frontal, parietal, occipital and temporal lobes and the cerebellum into two, so they're in fact paired, one on each side of the brain. There's also a deep fissure running along the side called the lateral fissure, which divides the frontal and parietal lobes from the temporal lobes. Here's the lateral fissure, and here is the central sulcus. Next we'll look at the meninges. The brain is covered by three protective layers. These are called the meninges. And in this diagram we can see them. There's a pia mater, the arachnoid mater and the dura mater. The pia mater is the part of the meninges closest to the brain. The dura mater is on the outside. This is what you'd first see if you looked into a brain, if you removed a part of the skull, and between them, holding them together, there is the arachnoid mater. An inflammation of the meninges is known as meningitis. The brain is bathed in something called cerebrospinal fluid, or CSF. And CSF is contained in the spinal cord and in the ventricles of the brain. So you can see here in blue the cerebrospinal fluid contained in the various ventricles, the lateral, the third and the fourth ventricles. There's an intraventricular foramen which connects the ventricles, and the cerebral aqueduct which carries the fluid down to the fourth ventricle and on down to the spinal cord. So effectively the brain and the spinal cord float in the cerebrospinal fluid, which as you can see surrounds the brain This offers the brain some sort of cushioning protection from movement and from blows to the body and the head. The brain and the spinal cord are composed of nervous tissue, and the basic functional unit here is the neuron, and the neuron consists of a body, an axon and dendrites, and then the axon terminals. And this coloured scanning electron micrograph shows what the neurons look like. Easier to see here in this diagram. At the top you've got a cell body which is connected to other neurons by the dendrites. Then you've got the axon, which is surrounded by myelin sheath. These are also known as Schwann cells, and you can see that this is discontinuous, and it's discontinuous at the nodes of Ranvier. And then at the bottom of the axon, at the opposite end from the body, are the axon terminals. These connect to the dendrites of the next neurons. So the dendrites receive incoming information from axon terminals. Axons, on the other hand, transmit information to the axon terminals. And information is transmitted along the neurons electrically through action potentials. And action potentials take place when neurons have an influx of sodium ions and become positive on the inside. This triggers an action potential which is transmitted along the nerve, and the action potentials are very quick due to saltatory conduction. They don't have to be transmitted the whole length continuously down the neuron. They jump between the nodes of Ranvier. We'll come back to looking at that. And here we can see what happens when the red sodium ions at the top, which is the outside of the axon, flood in. They're then pumped back out again. When they flood in, look at the millivolt meter on the right. At rest, there's a negative potential across the axon. When the sodium ions flood in, it becomes slightly positive. And this initiates the action potential, which then travels along the nerve. Watch it again. So here you can see an action potential being generated on the left, and it travels in one direction down the neuron. It's a bit difficult to see, but at every stage sodium is being pumped in, and in fact potassium is being pumped out at the points where the action potential is being generated. I mentioned saltatory conduction before. This is enabled by myelin sheaths and by the nodes of Ranvier. And essentially the action potential jumps between the nodes of Ranvier, and this speeds it up considerably. So that's what it looks like. Action potentials generated up at the cell body when the axons connecting to the dendrites stimulate them, and the action potential travels in one direction down the axon from the top down to the bottom, jumping between the nodes of Ranvier. The action potential only runs in one direction because it starts at one end of the nerve. In theory, the action potential could go in either direction, but there's a period called the refractive period during which an action potential can't be generated. So when an action potential is generated in one point, and that action at the top of the nerve, and the action potential then moves on, the part of the nerve where the action potential has been generated can't generate another one. So this ensures that the action potential doesn't move back up the nerve. It moves in one direction from the cell body down to the axon terminal. So let's take a look at the axon terminals. The axon terminal is the point at which a neuron passes information to the neurons with which it is connected. The electrical stimulus is transmitted chemically to the next cell by a chemical called a neurotransmitter, and the junction between the axon and the next cell, which is very small but real, is called the synapse. So here we see a synapse which is composed of the axon terminal and the synaptic cleft between the synapse and the postsynaptic membrane. The synapses are full of mitochondria which generate energy. These are used to pump the neurotransmitter back up into the cell once it's been released. So the neurotransmitter is released from vesicles which move towards the end of the axon terminal. The vesicles open up and pour out their contents into the synaptic cleft. They are picked up by receptors on the postsynaptic membrane, and that generates an action potential in the next nerve cell. Now it's important that the neurotransmitter, which happens to be called acetylcholine, is taken up again into the axon, or the signal would carry on indefinitely. This makes sure that the signal is very quick and once only until there's another stimulus on that nerve. So here in this diagram at the top we see the action potential travelling down. When it reaches the axon terminal, the vesicles containing the neurotransmitter acetylcholine move towards the membrane. Watch it again. They empty their contents into the synaptic cleft. They connect with receptors on the postsynaptic membrane, and that generates an action potential in the next nerve. So we've taken a look at the microanatomy of the nervous system, the neurons, and how they transmit signals between each other. Let's look a little bit more at the gross anatomy, and we can do this using the sensory homunculus. And here's the sensory homunculus. Essentially it's a grotesque looking man with very enlarged hands, lips and tongue, and other parts of the body which are very small in comparison. What this is showing you is how much of the brain is dedicated to sensing things from different parts of the body, and clearly we sense a lot through our hands but a lot less with other parts of the body, and we also sense a lot with our lips and our tongue. And we can also represent the homunculus on the cerebral hemispheres of the brain, and this shows in diagrammatic form the different parts of the cerebral hemisphere where the senses are picked up. We can also look at how much of the brain is dedicated to providing movement in the body using the motor homunculus. And here's the motor homunculus, and you'll notice that it's very similar. The parts of the body which are most sensitive are also the ones which are most mobile. And again, the motor homunculus can be demonstrated on the cerebral hemispheres of the brain. Let's return to having a look at the regions of the brain and what they do. First of all, I'll name them all. First the frontal lobe, then the primary motor cortex, and behind that the primary sensory cortex, then the parietal lobe, the temporal lobe, the occipital lobe, the cerebellum, and the brainstem. But what do the regions of the brain do? Well, first of all, the frontal lobe is responsible for decision making, problem solving, purposeful behaviour, consciousness and emotions. Any disturbance to the frontal lobe leads to very erratic and often socially unacceptable behaviour. The primary motor cortex, as the name suggests, regulates voluntary movements. And the primary sensory cortex processes sensory information received from the body. The parietal lobe receives and processes sensory information from the body. Letters and words form thoughts here, for example. Any damage there will affect someone's ability to speak. The temporal lobe regulates memory, emotions, hearing, language and learning. Interesting to note that the temporal lobe is affected in dementia. The occipital lobe processes information related to vision, so damage to the occipital lobe can interfere with sight. The cerebellum initiates and times movements, maintains posture and regulates force, steadiness and range of movements. You can still move and walk around if the cerebellum is damaged, but you do it again very erratically and often with too much force. And the brainstem connects the brain and the spinal cord, and here you have the areas which control heart rate, breathing, eating, blood pressure and so on. And the brainstem also gives rise to the cranial nerves, and we'll look at these next. So next we'll take a look at the cranial nerves. There are a lot of these emanating from the brainstem. They're called cranial nerves because they don't emanate from the spinal cord like most of the nerves in the body do. There are ways of memorising these and remembering them, but I don't think that's necessary here. So here's a list of all the cranial nerves: the olfactory, optic, oculomotor, trochlear, trigeminal, abducens, facial, vestibule, cochlear, glossopharyngeal, vagus, accessory and hypoglossal. The famous one here is the vagus nerve, which is the 10th of the cranial nerves. All the other ones are associated in one way or another with facial movements and facial sensing. The vagus nerve, on the other hand, supplies the rest of the body and plays a part in, for example, in digestion. If you do want to remember the cranial nerves, here's a mnemonic. An old Olympus towering tops, a Finn and German viewed some hops. All you've got to do is remember which nerve is which. As I say, I don't think this is essential information for you at this moment. The most important thing is, what do the cranial nerves do? As I've said, they mostly control functions of the head and neck. Eye movement, swallowing, smell, sound and so forth. Except the vagus, which plays a major role in the functions of the head and neck, and also provides parasympathetic fibres to nearly all thoracic and abdominal viscera. Now let's take a look at the peripheral nervous system in relation to the central nervous system. Here we see the central nervous system in pink in the middle, and the green on the right and the red on the left are the internal and external environments supplied by sensory and motor neurons. To the internal environment on the left we have the autonomic nervous system, which we'll come back to, and on the right we have the sensorosomatic nervous system. So the autonomic and the sensorosomatic nervous systems are part of the peripheral nervous system. So the peripheral nervous system has two divisions, the somatosensory and the autonomic. And the autonomic nervous system is further subdivided into the sympathetic and the parasympathetic system. Let's take a look at those. This is how these are normally represented, with the parasympathetic on the left and the sympathetic on the right. If you take a quick look at the sort of things the sympathetic system does, dilates the pupils, inhibits saliva flow, accelerates the heartbeat, dilates the bronchi, inhibits digestion, and so forth. Essentially, the sympathetic nervous system gets us ready for action, fight or flight reaction as it's sometimes called. Parasympathetic system generally has the opposite effect. So what are the special senses? Well, there's sight, smell, hearing, touch, and taste. Let's take a look at each of these in turn. First of all, sight. The organ of sight is the eye. It's a very complex organ, and it looks a bit like this. So here's a diagram of the eye, and the light in the particular diagram we see here is travelling from left to right. If we follow the path, the light penetrates the cornea, which should be clear in normal health. Sometimes gets cloudy in older age, that's called cataract. The light then travels through the aqueous humour and then passes through the lens, and the pupil is the area which is controlled by the iris, and the pupil is the area through which the light passes into the lens, and it varies in size depending on the light conditions. So light passes through the lens. The lens is shaped and supplied with muscles such that it can focus the light in different ways depending on the distance you're looking at. For example, it focuses differently if you're reading or looking across the road. The light then travels through the vitreous humour and hits the optic disc or the optic nerve, and there the light is transmitted to the brain. So the organ of sight is the eye. Light is captured by the eye and transmitted to the brain for processing into images. We can see that here. Because of the way light is transmitted, images are transmitted to the back of the eye, and they are there upside down. However, the brain then inverts the images so you can see them the correct way up. And the retina contains photoreceptor cells. These are cells which, due to their chemical composition, react with light. This is just a cross section of that part of the eye, at the back where the light reacts, showing the pigmented epithelium photoreceptor cells and the synaptic terminals. The orange arrow is the light coming into the eye. There are two types of photoreceptor cell, rods and cones, and these serve different functions. The rods are active in dim light and the cones are active in bright light. The photoreceptor cells contain visual pigments, 4 different kinds: blue, green and red, capture different parts of the light spectrum, and monochrome for black and white. So essentially, a broad spectrum of light is covered by these different photoreceptor cells. The organ for sensing smell is the nose. And in this cross section of the head you can see the nasal cavity above the mouth. And here we see the olfactory nerve, and in fact the olfactory nerve is connected directly, very directly, to the brain. The organ of hearing is the ear. Here we see a diagram of the ear. From left to right, sound travels in. It's captured by the pinna, or the ear lobe. This is the visible part of the ear. Travels down the external auditory meatus to the tympanic membrane. There the sound is transmitted to the cochlea by 3 small bones, the malleus incus and the stapes, which move. Then the sound is transmitted via the auditory nerve to the brain. So sound is transmitted from the outer ear through the middle ear to the inner ear and to the brain via the auditory nerve. Here we see sound coming in and the bones of the inner ear moving, sound travelling around the cochlea and on through the auditory nerve. Watch that again. Next, touch, and the organ of touch is the skin. We will look at skin in more detail in subsequent sessions on protection, but let's have a quick look at it here. Here's a cross section of the skin showing a hair shaft in its hair follicle with the erector pillae muscle, which makes the hair stand up when you have goosebumps. There's oil glands called sebaceous gland which lubricate the hair shaft, and there's also sweat glands and various other corpuscles at the bottom. The skin's divided into 3 layers: the outer layer, the epidermis, then the dermis containing these structures, and then the hypodermis below it, which is a fatty layer. The skin contains several receptors for cold, touch, pain and pressure. And here diagrammatically we see them. There are thermoreceptors sensing heat or cold, Meissner's corpuscles sensing touch, nociceptors which sense pain, and then the Purkinjean corpuscles which sense pressure. Let's have a quick look at pain. This is a sensory process. It's also part of our protective mechanism. And let's have a look at the gate control theory of pain. It's a very crude theory about pain, but it's been very effective in helping us to understand how to treat pain. It's clearly been refined since its original conception by Melzack and Wall. But let's just have a look and see what the basic components of the gate control theory of pain are. If you have an event or an injury, then there are gates in the spinal cord, and these gates can be opened or closed. That's the basic theory. If the gate's open, you experience pain. If it's closed, you don't. And various factors can have an influence on opening or closing the gates. So this diagram shows the gate control theory very, very crudely. You can hurt yourself by dropping something on your foot very directly, sending out a signal of pain to the spine, or you can have a more dull sort of ache, like a muscular ache or a backache. The extent to which these are sensed by the brain depends on whether you are stressed or in a low mood. If you're frightened, for example, you tend to feel more pain. If you're being distracted or enjoying yourself, or there are endorphins released in your body from doing something you enjoy, like exercise, for example, these tend to close the gate. And the gate control theory can be exploited pharmacologically because drugs like opioids act on the gates to close them. That's the gate control theory, very crudely. Next, taste. The organ of taste is the tongue, but we need an intact sense of smell to contribute to the sense of taste. Most of what we taste is actually contributed by our sense of smell. Taste is enabled by taste buds, of which there are approximately 10,000 on the tongue. Here's a tongue, and those are the taste buds, the little raised areas on the tongue. If you magnify a taste bud, it looks like that. There are different types of taste bud, and they are distributed differently over the tongue. The circumvallate at the back, and the foliate at the side, and the fungiform at the front. And essentially on the right you have the surface of the tongue reacting with molecules which provide taste, and it sends a signal to the brain which conveys the sense of taste. Different regions of the tongue sense different tastes. Therefore we tend to taste sweet substances at the front, bitter substances at the back, and sour substances at the side, and the centre of the tongue doesn't have so many taste buds. So finally, the nervous and sensory systems are composed of the central nervous system, the peripheral nervous system, and the special senses. So to end this session, I'm going to ask you some questions. 1. What is the collective name for the protective layers of the brain? What are they each called? 2. What fluid is found in brain and spinal column? Where is this found in the brain? And state 2 functions of this fluid. 3. Name the main features of the neuron. 4. Explain what happens in an action potential. 5. Explain what happens at a synapse. 6. Explain saltatory conduction. 7. Name the 8 regions of the brain. Half a mark each for these. 8. What do cranial nerves mainly do? 9. Name the 2 divisions of the peripheral nervous system and the subdivisions of the division concerned with involuntary actions. 10. In addition to sight, what are the other special senses? 11. What 4 structures of the eye does light have to travel through to reach the optic nerve? 12. What are the 3 bones of the inner ear and what are they called collectively? What 4 types of light do the pigments in the photoreceptor cells, the rods and the cones, detect? 14. What is the first structure that a sound wave comes into contact with when it passes through the external ear? And what are the 3 ossicles of the ear called? What do the 4 sense organs of the skin sense? Which part of the tongue senses taste? And what are the 3 tastes? Thank you very much for listening. This is my email if you have any questions or comments. All the answers to the questions can be found in my blog, 4 Things About, a simple approach to anatomy and physiology, on the entry, the nervous and sensory system, and the special senses. Once again, thank you very much for listening.